So today we're going to talk about the denaturation of eggs. So proteins are large molecules that are comprised of different amino acids. They are constantly folded upon one another, which gives them their tight structure. And what you're seeing at the bottom here is an example of one of the proteins that is found in eggs. And if you look closely, you can see some of the coils that are around the protein. And it's kind of a whole bunch of stuff going on. And a lot of that is because it's very compact and there's a lot of folding that is occurring. If you were to straighten it out, it'd be very, very long. So one of the reasons why proteins look like that and have that such a tight folding appearance is because of their disulfide bonds. So what happens is that there is an amino acid called cysteine, which has a thiol functional group. And what happens is that it goes through a process called oxidative folding, which is a redox reaction. And what happens is that the cysteine bonds, the thiol bonds, become oxidized, and that will form the disulfide bridges or disulfide bonds throughout the protein. And these bonds are critical for the 3D structure of proteins, and that those bonds allow them to give the protein its distinct shape and function. So what you're seeing down below on the left is a two cysteine amino acids that have a disulfide bond right at the sulfur. They're connected to each other. And the one on the right is an example of what you would see in a protein. So you can see how it winds around, and then all around it has those disulfide bonds. And in a protein, there's going to be an extreme amount of those bonds, and that's what really helps give the protein its distinct shape and function and make it strong. So today what we're going to be doing and observing is the denaturation of those of the protein. And what that really means is that those disulfide bonds or those disulfide bridges are going to be those bonds are going to be broken up. So when there's extreme changes in pH or in temperature, that causes the bonds, those disulfide bonds, to break apart. And whenever those bonds break apart, it causes a conformational change in the protein. So those disulfide bonds really are important for the protein. And whenever those bonds are broken, it really changes the shape of it, as you can see right here. So when that denaturation process happens, the shape of the protein and the shape of the protein changes, and there are new properties that are observed because you're changing um, you're breaking some of the chemical bonds. So and that happens a lot with other chemicals. When you alter the chemical bonds, you can either raise or lower the boiling point or melting point of the chemical, so or the molecule. So um, with the distinct shape of the new shape of the protein, you can see certain types of solubility and different enzymes, and how the enzyme will react to that new denatured protein. And in the egg, um, one of the observations of this occurring is that when you start with the egg, it starts as this clear and transparent liquid, the egg white, and it goes to a kind of a solid, a white pasty solid. And that occurs because of the denaturation of the protein. And denaturation happens. This happens every time you are served eggs for breakfast, and is why if you ever had a raw egg, tastes way better than, or if you had a cooked egg, it'd be way better than a raw egg. I mean, I don't know anyone that likes to eat raw eggs, and this is important because it's served as a food source for a lot of different animals and humans, and it's very tasty. So that's one thing. And another thing is that during the Middle Ages, the denaturation of these eggs also provided them with the ability to make the typical middle-aged book, which is what's called a illuminated manuscript. And the writing material that was used to make these books were use denatured yolk to do that. So I have the pan. I'm going to place the egg inside of the pan, and let's see what happens. So I'm gonna pause it for a second. So as you can see, the liquid, or the egg white, is very, you can't really notice it. It's very clear and transparent. So let's turn it on and let's see what happens as we cook this egg. All right, so you can start to see it hasn't happened all over, but this is just the beginning of it. The that white clear substance I was saying, you can see the protein is beginning to form or get deformed. Those bonds are getting to be broken. You can see that it's starting to change its color, and it goes from a clear to like a white, almost liquid-like substance when it's almost done. So let me have this going for a while and see what exactly happens at the end. So now as it's getting more quick, you can see how it's a lot more solid, and it's becoming it's becoming a lot more solid than what it was before. It's not as liquid as it was before. It's really changing its shape. So I got let it cook for a while, and I extracted the egg, and you can see I broke the yolk there, but you can see that it's very different than what it was before. And you can see that it actually underwent a change, and this happens because the proteins were denatured. And one of the benefits, at least for food, what this does is it allows the your enzymes to actually digest some of the proteins inside of this, because in its actual protein state, when it's not denatured, sometimes enzymes can't do that. So some food actually becomes your body is able to extract or digest more of it when it's cooked versus it being raw. So yeah, it's very different than what it was before. You can see the change in the egg whites, and yeah, so that's the denaturation of an egg.